Now that you know a little bit about why a cell is going to respond, which is either in response to a signal or responding to another cell, I want to describe to you how a cell actually responds to those signals once the cell's been induced to respond to its particular environment. So what you'll notice here is we've got our cell surface receptor. So here is the cell surface receptor, again, nested in the phospholipid bilayer, and here it is in its unactivated state. Upon binding to the signaling molecule, you'll see that the cell surface receptor undergoes a conformational change. So here it has this kind of hooked conformation, and now we see that that shape is different. And what that's going to do is going to initiate a signaling cascade, or sometimes we call this signal transduction. So the message from the outside of the cell is now transduced or sent within the cell until that ultimately is going to cause the cell itself to respond in an appropriate way based upon that extracellular ligand. So the three most common responses that one will see are either you'll see enzymatic activity, so we're going to have perhaps altered metabolism in response to that cell signal. You might see structural proteins getting modified, so perhaps the cell is going to move towards or away something in response to that signal. Or lastly, the one we'll spend a lot of time talking about is the activation of target genes. So based upon that cell signal, we have a signaling cascade, and that's going to turn on or turn off a whole host of genes based upon what that extracellular signal is. And those are our three most common cellular responses. All right, so that basically summarizes section 8.1. So we talked through the reasons for cell signaling. I put a bunch of videos up on the different models of cell signaling, and then I've just highlighted what does that process actually look like. So we're going to skip section 8.2, receptor activation, in your book, and we're going to go ahead and jump into section 8.3. For all of the different methods that we're going to talk about, they have the same overarching series of events that's taking place, and that includes the following. So we are going to have a hormone, and another word for a hormone is a signaling molecule or a ligand. So you'll hear these terms used throughout the rest of the chapter. And that hormone or ligand signaling molecule is highly specific for a particular type of cell surface receptor. And here we have that cell surface receptor in an inactivated state. It is waiting to bind to a particular signaling molecule. Upon binding of that ligand, which has high specificity for that particular receptor, the receptor itself is going to undergo a conformational change, which will lead to a signaling cascade. Once the ligand dissociates from the receptor itself will go back to that unactivated state. And so we'll keep coming back to these terms, so make sure you feel comfortable identifying what is the signaling molecule and what is the receptor, what is the signaling events that take place within the cell, and then ultimately what is the particular cellular response. So we are going to skip section 8.2 of your textbook on receptor activation, and I'm going to give you an overview of section 8.3 on the different types of cell surface receptors, and then we will go deeply into section 8.5, which is actually enzyme-linked receptors, and then section 8.6, which are the G protein coupled receptors. But right now I'm just going to introduce all those kind of big picture. So with regards to our cell surface receptors, these are going to be enzyme-linked, G-protein-coupled receptors, or ligand-gated ion channels. So let's start with our enzyme-linked receptors. So these are activated receptors that have enzymatic activity once they have bound to their ligand. And so we have two very important domains that I need to point out. The first is the extracellular domain. So this is outside of that phospholipid bilayer. And this is what's going to bind to that ligand or signal, hormone, etc. Upon ligand binding, we're going to have a conformational change in the receptor, and that conformational change is going to lead to catalytic activity. So this intracellular portion is going to be what drives the signaling cascade within the cell. For the most part, what these are going to be involved in having kinase activities. And what is kinase? That indicates that something got phosphorylated. So what you can see here is we've got a target protein. This is a inactive, unphosphorylated protein. Usually it's inactive. And upon binding of the extracellular ligand and the conformational change, the catalytic domain of our cell surface receptor is going to result in the phosphorylation of this downstream target protein. So you'll see ATP became ADP, and that phosphate group got transferred to the target, and that was all mediated via that cell surface receptor, which is responding to the extracellular ligand. When I say kinase, what exactly does that mean? It means that we had a protein that after kinase activity got phosphorylated. And there's certain types of amino acids in proteins that can get phosphorylated. Serines, threonines, tyrosines, these all are able to have a phosphate group attached onto it. So the addition of that phosphate group is going to modify the folding of the protein. And if you modify folding, you're going to modify structure, and structure determines function. So by adding that phosphate group, you're going to change how that particular target protein behaves. Okay, and it depends. Sometimes we're turning a protein on, sometimes phosphorylating it actually turns it off. So you'll just have to become familiar with what types of signaling cascades we're talking about. The second class of cell surface receptor proteins that we will explore are the G protein coupled receptors. So the G protein coupled receptors, that here we are in the phospholipid bilayer. Here's our extracellular portion, the intracellular portion you can see here. The extracellular portion again is going to be that region that binds to the ligand. And upon ligand binding, we're going to have a conformational change in the receptor. Now these are a little bit different. They're not enzyme linked, so they're not causing the phosphorylation of a particular target. But here we've got G proteins. We've got a bunch of different G proteins. We call this alpha, beta, and gamma, and they are bound currently to GDP. But upon ligand binding and that conformational change, you get recruitment of these inactive G proteins to the receptor itself. And upon interaction with the G protein coupled receptor, you're going to get release of the GDP and binding of GTP, guanosine triphosphate, and that is going to lead to dissociation of that G protein with all of its subunits. So now the alpha subunit is liberated from the beta and the gamma subgroups, and now this is going to drive a whole series of events within the cell. Once ligand dissociates, eventually you're going to get dissociation of this GTP and recruitment of the GDP, and these three G protein subunits will interact with each other once again, and we'll have this unactivated pathway waiting to bind ligand. Now, a third type of cell surface receptor are ligand-gated ion channels, which we've actually talked about before. And this is a little bit more simple picture. So upon ligand binding, the channel can open. Sometimes it's the opposite. Sometimes upon ligand binding, the channel will actually close. But based upon the description and the pictures, you can figure out which way it is. So here we've got ligand binding. The channel opens, and that allows for the influx of a particular solute, an ion, etc., that normally could not enter through the phospholipid bilayer, but now through that ligand-gated ion channel, we can get a massive, quick influx of that particular solute from the extracellular environment into the intracellular environment. So those are going to be our three big classes of cell surface receptors, and I briefly want to talk about section 8.4, which are our intracellular receptors. Okay, our intracellular receptors are going to be binding to a bunch of these hormones and hormone derivatives of cholesterol that are going to be able to make their way through the phospholipid bilayer. So if you look at all these structures, these are predominantly hydrocarbons, cyclic hydrocarbons. Sometimes there's a hydroxyl group, but for the most part, these are going to be able to permeabilize through that phospholipid bilayer. So they actually do not need a cell surface receptor in order to mediate their response because they can permeate the cell on their own. So these signaling molecules will make their way through the phospholipid bilayer, and they actually go all the way into the nucleus itself. So we call the targets of these types of signaling molecules nuclear hormone receptors, and because they're residing in the nucleus. And so here we have that signaling molecule, for instance, estrogen. Estrogen is able to go through the phospholipid bilayer all the way through the nuclear pore. It then finds its target inside the nucleus, which is the estrogen receptor, and estrogen receptors are going to dimerize, and they will then sit on within the promoter region of a particular target gene and turn on or turn off specific target genes, ultimately making mRNA and then proteins that can respond to that cell signal, the estrogen. This is what I actually worked on through grad school. I worked on intracellular receptors. I worked on nuclear hormone receptors, specifically a bile acid receptor. And that's what I spent 7 years of my life studying in grad school. So I did want to include it here.